Hi, I'm Xiaoxuan from Allen Institute. Today I'll talk about subnetworks mediating feed-forward and feedback processes in mouse visual cortex reviewed by multi-area recordings and the large-scale functional network analysis. So the visual system is organized hierarchically with extensive feed-forward and feedback connections forming a highly recurrent network. For example, here I'm showing a graphic representation of the monkey visual areas with different boxes represent the different cortical area, organized according to their anatomical hierarchy from bottom to up. V1 sits at the bottom of the hierarchy. The black lines indicate bidirectional connections between areas. So you can see it's both hierarchical and recurrent. Recently, a study found similar structure in mouse visual cortex based on anatomical tract tracing experiment. Here is a simplified representation of the graph in mouse visual cortex. Different circles represent different mouse visual areas, again organized according to the anatomical hierarchy score, with V1 sits at the bottom of the hierarchy, and you may notice it sends output and receive input from all higher visual areas. So this network is also both hierarchical and recurrent. So given this structure, the hierarchical and recurrent anatomical structure, we wonder how information flow in this network. And we address this question by building a large-scale recording platform to record the spiking activity of single units from six interconnected visual areas simultaneously in awake mouse. And we used Neuropixels probes, which has been introduced in Churchland's talk, which is a high-density recording probe, can sample around 100 units or more than 100 units simultaneously. And we built a rig to record 6 of these probes simultaneously, while the mouse is head fixed on the rig, free to run on running wheel and watching visual stimuli. The 6 probes will be inserted vertically through the 6 visual areas I introduced in the mouse connectivity diagram. V1 and the 5 high visual areas surrounding it. And because it's a linear probe, this will provide us a multi-area, multi-layer network. We targeted overlapping receptive field to maximize measurable functional interaction using retinotopic map obtained from wide field imaging. Here is an example of the retinotopic map. And we define the cortical layers or depths according to current source density. And in general, we separate the layer into superficial, middle, and deep layers. So all the results I'm going to show you today are based on using visual stimuli, drifting grating visual stimuli. Here is a raster plot showing the spiking activity from simultaneously recorded units from the 6 visual areas. On average, we can get more than 600 cortical units in each mouse. We quantified the functional interaction between each a pair of neurons using cross-correlation by computing a cross-correlogram based on their spectrings. Basically, the cross-correlogram, or CCG, measures the probability of one neuron generate a spike with different time lags relative to each spike in the reference spectrum. So to give you an intuitive understanding of what this matrix means, so if you have 2 spectra, X1 and X2, X1 is from the reference spec or source neuron, X2 is the target neuron, and if they have the exact same spec times, you will get a CCG with a peak at 0 time lag. If the X2 spectrum is consistently later than spikes in X1, you will get a peak with positive time lag. And if it's consistently earlier than the source area, you will have a negative peak at a negative time lag. So basically, CCG measures the relative timing between 2 spectrums. It reflects not only the strength of connection, but also the directionality of connection. That's why we use this metric. It is important to note that the cross-correlogram reflects the correlation in a range of timescales, but we are more interested in signal transmission happen at the fast time scale. So we use a jitter correction method to remove slow timescale correlations. And by subtracting the CCG calculated based on jittered spikes from the original CCG, So basically, the remaining jitter-corrected CCG will reflect brief timescale correlations within the jitter window, which we chose to be 25 milliseconds. So here on the right, I'm showing an example jitter-corrected CCG between one pair of units. You can see it has both. It has a peak with certain time delay relative to 0, reflecting correlation strength and the directionality. So to use a single value to quantify this magnitude of functional correlation and directionality, we just take the integrated difference between 13 millisecond time window before and after 0, and construct, if we do this for all pairs of CCGs, we will get a single cell resolution functional connectivity matrix with each pixel representing the magnitude of each pixel represents the functional correlation strength, and the sign of each pixel represents the directionality. So in this matrix, the axes are units recorded simultaneously in one experiment, organized according to their depths and the areas. As you can see, there are some non-random structure in this matrix, and we want to analyze this non-random structure in this matrix. So we take the connectivity pattern from each source neuron to the network as features and did clustering on this connectivity matrix. We found 3 different clusters. Cluster 1 basically dominant with weak connections. Cluster 2 is dominated by mostly positive connections, and the cluster 3 is dominated by negative connections. To give you an intuitive understanding of what those connectivity pattern means, if you take a source neuron from this cluster 2, you see mostly positive connection to the network. That means this neuron is leading or driving the network activity. And if you take source neuron from cluster 3, you see mostly negative connections. Like it means this neuron is mostly following the network activity or driven by the network. So this separation into 3 clusters is very consistent across mice. Here I'm showing 6 more examples showing the separation of 3 clusters. And I quantified the total connectivity in each cluster. Here I'm showing, in total we have 19 mice. So basically cluster 1, you see mostly very weak connection. Cluster 2, you see significant positive connection to the network, and the cluster 3 you see significant and negative connection. And from now on, I'm going to ignore cluster 1 and call cluster 2 as driver neurons and cluster 3 as driven neurons. So you see there are interesting structure in the connectivity matrix. We want to visualize the structures. We use a graph to represent the structure. So each node in this graph represents a unit recorded simultaneously in one experiment. And it is organized according to their cortical depths from superficial to deep, clockwise in each area. And I'm going to draw a directed graph, which means red color indicate connection from source to target, and the blue indicating connection back to source. And here I'm going to use source area as in V1 as an example. Now I'm plotting the subnetwork between driver neurons and in V1 in the source area and the driven neurons in all other high visual areas. You can see it's mostly dominated by positive connection, meaning the driver neuron is leading the network activity. And here I'm plotting the subnetwork between driven neurons in V1 and the driver neurons in high visual areas. You can see the information is flowing back to V1. So clearly these two subnetworks are separable, and the driver and the driven neurons also showed layer dependency in the source area. So this is V1. How about other high visual areas? Here is another example in the same mouse. If I'm using LM, a high visual area in mouse visual cortex as the source area. And you can see clearly we can still with the method, we separated out the input and output to the area, and they have clear layer dependency. To quantify the layer dependency and separate it into the 3 layers I just mentioned in my introduction, we see a bias in the distribution. So the driver neurons are more biased to reside in superficial and middle layers, while driven neurons are more in the deep layers. More interestingly, their proportion also changed as a function of the anatomical hierarchy, with the driver neurons decrease as along the hierarchy, while driven neurons increase along the hierarchy. So previously, in the anatomical study showed the proportion of supragranular feed-forward projecting neurons decrease along the hierarchy, which agree with our trend of the driver neurons. And along with the directionality of the driver neurons, we think driver neurons participate more in the feed-forward process, while driven could participate more in the feedback process. So I separated out two groups of neurons based on their functional connectivity to the network. Now we wonder whether they have some signature in other domain. So we looked at their temporal dynamics in each single neuron. And here I'm plotting the population average PSDH for driver group and the driven group. As you can see, the driver neurons are more transient earlier, and the driven group is later and more sustained, which agree with their potential role participate in feed-forward and feedback process. So these are single neuron property. We also find that as a population, these two groups of neurons are also different. Here I'm showing an example. This is the population response precision to visual input in single trials. And on the left, I'm showing the raster plot of a population of driver ensemble in one single trial. Each line is one neuron. The black dot represents spikes. The red cross represents the time to first spike. And similarly for the driven ensemble. And you can already visually see the time to first spike in the driver ensemble is more precise in response to visual input compared to the driven ensemble. And I quantify the distribution as the standard deviation of the distribution. Across all mice I have, the driver neurons are more temporally precise and more time-locked to visual input, which also, again, agree with its potential role to sincerely carry visual input. So today, I just showed you we found two distinct ensembles distributed across visual areas that could participate different in feed-forward and feedback process. And this is suggested by their direction of functional connectivity, their distribution across layers, and along the anatomical hierarchy, the difference in their temporal dynamics, and their population response precision to visual stimulus input. And the one thing I want to emphasize is, in Allen, we emphasize on open science, and so this data I presented today will be available in October this year. So I want to thank my advisor, Shawn and our chief scientist, Christoph, for their support in this line of research, and my important collaborator, Siegle, for developing the multi-probe recording platform, and our fantastic eFace operation team to collect the data, Séverine, Durand, Greg, Heller, and Tamina. And there's another part of research I don't have time to show you today is to predict neurodynamics based on the connectivity matrix. This is in collaboration with Yazan, Billeh, and Anton. Thank you for your attention. Now I can take questions.
Very cool. We have time for a number of questions.
There.
This is beautiful data, thanks. I was wondering if you looked at whether these roles that these neurons play, driver versus driven, how stable they are across different brain states, spontaneous versus evoked, or other ways of defining different brain states.
Yeah, actually there are many things I don't have time to show you. So one line of research is we actually compared across different visual stimuli. Like we have drifting grading, we have natural movie, and also the spontaneous condition. So basically, if you consider this network, it's like if you consider the structure of network, why a recurrent network has a hierarchy. So it's a hierarchical and recurrent, and the input, the information drive into the hierarchy, forming a flow of information. Like it has some latency, and if you use different type of visual stimuli, potentially different brain state, the strength of this feed-forward flow will be different. It's really stimulus dependable and state dependable. So basically what we are showing is only one state of the functional network. This functional network is very dynamic. The information flow really depends on external stimuli and internal brain states. Thank you for the question.
Hi, thanks for your talk. I was wondering if you guys have started analyzing your LFP data, and also if you've looked at inhibition and which layers provide, driven versus driver inhibition, and if that's different feed-forward feedback and so on and so forth.
Yeah, actually, yeah, we are. I totally understand where the question is coming from, and we are looking at the LFP. We are actually looking. Also, not only look at power, but also spec field coherence and the granular causality in the frequency domain to look at the in-out between cortical, different areas and also across layer, because this is very important information. Yeah, we can talk more if you're interested.
Two technical questions. You've considered integral from -13 to +13 milliseconds. Is it possible to get more detailed, temporally detailed information by changing this window? And second, the entire analysis is based on second-order correlations. Have you thought about extending that? I mean, it's a network. You might expect that there are modulatory effects, that there are higher orders that are important for the dynamics. And can you actually talk about functional connectivity by just considering second order?
So to address the first question, the reason we use 13 millisecond window is because in the jitter correction, we use the 25 jitter window. So that's basically almost half of it. And if we basically, the jitter method capture different, remove different time scale of correlation. And if you're interested in more finer time per cycle correlation, we can use 10 milliseconds. It's just it's more related to relevant to the monosynaptic connection. And if we use larger jitter window, it have more slow time scale correlation related to noise correlation, like the internal variance. So that's definitely very interesting. To address your second question, the higher order functional interactions. So currently, for my functional analysis, I just use the pairwise, but I was looking at dimensionality reduction and the manifold representation in the more higher dimensional domain. I think that's very, very interesting, and I was actually hoping when we get this a graph based on pairwise analysis, we can already start to look at higher order in the graph. Do you think it's possible? Yeah. Yeah, but it's very interesting, yeah.
Any other questions? I have one. How does your functional measurement compare to anatomical estimates about the strength of the connections?
Actually, we are writing a manuscript on that direction. So we actually, the anatomical data is actually produced in the Allen Institute. So we have the full access to the anatomical data. And anatomically, they showed this, the hierarchical directionality. And if I constrain my functional analysis to monosynaptic connections, and I can basically generate a similar hierarchical map according to, similar to the anatomical structure. And you can look forward to our manuscript.
Okay. Well, let's thank all the speakers again.
Thank you. Thank you.